I've been working on Alzheimer's disease since 1984. I'm at the University of Southern California. I'm a member of the Department of Pathology, and I'm a neuropathologist, and that we are the ones who give the definitive diagnosis. The clinicians are very good, but we're the ones with the gold standard diagnosis. I became actively involved with research in Alzheimer's disease in 1984 when we were granted from the National Institute of Aging a large center grant on Alzheimer's disease, which allows us to follow patients longitudinally, and then we have people to sign for autopsy, and at the time of death we have a system that brings in the deceased rapidly so that the tissue can be used to study the disease as well as make the diagnosis. The clinicians are very good, but as I say, we have the definitive diagnosis. They're wrong sometimes, and it's very important for families to know exactly what the deceased has. We have, these patients are followed longitudinally, and I'll go over a little bit how this is done and what are some of the hallmarks of the disease that allow us to make this diagnosis. Now, the National Institute of Aging has, since we were the first of 5 centers, among 5 centers to be established, now there's close to 30 in the country. And this has been very important because it gives us a longitudinal look at what's happening with the patient. They have neuropsych battery that allows us to find out about how this is behaviorally connected to the changes in the brain. And the tissue is available also, not only to make the diagnosis, but there's no good research model that's perfect yet available after all these years. There are mouse models of parts of the disease, and I'll explain that briefly. But none of them really give us the full look at what's actually happening, both at the cell and the molecular level. There's some genetics that have been done, and nowadays we have so many patients that have been enrolled that a significant number can be evaluated and genetic analysis applied, and that will be significant. And because the centers combine their data, and this is done, we have a statistical branch to do this for national receipt of the data from all the centers, that we get significant information, and it isn't just hearsay then. And that's part of the genetic analysis done by scientists who do just that. And everybody would like to know, is there one gene or one enzyme? It's not that way with Alzheimer's. It's a disease that is has multiple causes, multiple genetic contributions. There are risk factors involved, so it's not so straightforward. This is why it's been taking so long to really understand what is going on in the disease. And I became involved in it in 1984. I was the co-director of this center and have long after that been co-director now and done the diagnosis neuropathologically. Now I do neuropathological diagnosis as well, and have continued then to be able to do research on the human brain tissue as well as in disease models, for some answers at least, in my own laboratory. And I'm continuing to do that. I'm still working full-time. We have several NIH grants that allow me to do that. And I will show you a little of the work we're doing, but just give you a little bit of the information of some of the terms and how the patients are evaluated and what are some of the problems and why is it taking so long. Everybody wants to know, what can we do right away? How can we help? It's been difficult, and there's many, many people in this very well organized system that are trying to get this kind of help to everyone. Okay, that's me about 3 years ago. And this was published in LA Weekly. Interesting people in Los Angeles in 2009. It was Jonathan Gold, the critic, the food critic, who I knew, and he decided to come and see brain tissues. He's very curious. He got a Pulitzer Prize about 2 weeks after this was taken. I don't know if any of you know about Jonathan Gold. There's a movie called City of Gold now about him, but he did this, and it's not to be eaten. Needless to say.
Except in France.
No.
Except in France.
But not human brain. Besides, the cholesterol is too high. So, I'm staring at that brain, but I've seen many and have expertise in the diagnosis of this. So the questions are, where and when does the disease process begin? It goes on for many years. You know, this is a late onset disease. It's very long and chronic in its course. And the question is it reversible early? And that's one of the things NIH has inspired most of us, with funding, of course, the inspiration help, to do. And it's important because structural changes occur in the brain that can't be reversed. So the goal is to catch it and detect it as early as possible. That's the first goal. The second goal would be to do something about it. And if we know what's happening, then we have targets to aim for. What is the toxic molecule and where does it act? I think people who understand and know about this disease know about some of the players or bad players in it. And why are only some nerve cells, neurons, vulnerable? So there's selective vulnerability. Some cells are vulnerable and other cells are not in the same region. So that's a question that many of us have targeted in our research efforts. Does the affected neuron die? Well, we don't know because we only can catch it when we get it. But obviously some of the changes are there in the neurons, and it still is alive. It's still there. It hasn't disappeared. So these are some of the questions. And there are many, many other questions. I'll just try to give you a simple format to digest. So what are the clinical features and when is it detectable? Now everybody says there's aging and you can't remember it. That may be true. It's complex, but there's progressive loss of memory and cognitive functions resulting in dementia. Now dementia is loss of ability to have a normal cognitive function. And many of the neuropsychological tests that are given test that. And they relate to function in different parts of the brain that we know are affected neuropathologically. The time from onset of symptoms until death averages 8 years. And we've had some patients come in after, who are signed up and then pass away, come in after 14 or 15 years and some earlier. But it's usually an average around 8 years. It can range quite a bit longer. And that's of great importance to families. What do families, how do they deal with this when people are demented? Now, I want to say one other thing about the term dementia. There's a pie chart I use to teach medical students and residents and all about this. And the biggest portion of that related to cause of dementia is Alzheimer's disease. Dementia can occur in strokes or vascular problems in the brain. It's associated with other neurologic diseases such as Parkinsonism. But it's Alzheimer's disease that is the main cause or associated mainly with dementia. So when people talk about dementia, it isn't just Alzheimer's, it's many other things. And the doctors have to, the neurologists have to rule that out. Because some of these are fixable, like subdural bleeding, subdural hematoma, could cause loss of cognitive function, but that can be reversed by removal of the blood clot. depression is associated with it. So we have to consider all of those things. And then I remember one case that I had to diagnose, and they said, you get a social history as a patient drinking, and everyone there said no, but we couldn't find any Alzheimer's or anything else. And then they revealed the guy drank a pint a day or some large amount. So we're on the alert to do a full differential. The clinicians do this. And they also can do diagnoses using imaging, and there are markers that bind, and I'll tell you about that as I go through this. So here's the brain postmortem, and it's marked with all these little letters on it, and those are functional regions of the brain. So right here is the frontal lobe. Here's occipital lobe. This is where vision is. You can't see the region here. It's medially, internally. This is for higher order functioning in the frontal lobe. And the frontal lobe, there is involvement selectively of parts of the brain here. This is a normal brain, and it's marked, but I'll show you an abnormal one. Here's temporal lobe, and this is where hearing and speech is important. This is the sensory region for if you touch something, it's somatosensory. This is motor, and this is involved not only, it's not involved much in Alzheimer's, but it can be involved in motor neuron disease, and that's different. That's like Lou Gehrig's disease that has this part can be involved. And so, and there's a part inside here called the hippocampus, and I'll show you that. That's where memories are first encoded, and that's the first part where symptoms show in the disease. So there's a progression of changes in here that tell you where the patient is along the disease path. And this is an actual Alzheimer's brain, and here is the frontal. It's looking at the top down. Here's occipital, here's cerebellum, the back of the brain, and here is the motor and the somatosensory areas here. They're nice and plump, and there isn't much space, and the hills and valleys are the gyri and sulci. The gyri are plump, the sulci are the little valleys. These are widened in Alzheimer's. This area, you can see, is much smaller in the frontal area. That's involved in this disease. And other dementing diseases have other patterns, and the function of those regions is taken in consideration by the neurologist and by the imaging results. And there's a pattern that fits. So this fits with Alzheimer's. This area is involved. These are much, much less involved. This is parietal lobe. That's involved. That's an area that it includes the, it connects a lot of the, this is here, sensory modalities together, like vision and touch are sort of put together here. So this part is actually very important, and one of the areas very much involved in the disease. This is the other side that's also involved, but we've left on the thickened leptomeninges, the covering of the brain.
I couldn't figure out what that was.
That's what that is. So we've taken it off over here. But it's important to know where the pattern is of this disease. It's a neuronal disease. It doesn't involve other cells much in the brain, the glia, which are the supportive cells.